In this video, we're going to discuss the pyruvate dehydrogenase complex. So we know that glucose passes through the glycolysis pathway and ultimately produces the molecule pyruvate, or actually two of those. Now, pyruvate then will enter into the citric acid cycle where it's further oxidized, and those electrons, as we'll see later, will be used to make ATP. But there's a very important precursor step. In order for pyruvate to enter into the citric acid cycle, it must first be converted into a molecule called acetyl coenzyme A. So this precursor step, this precursor to the citric acid cycle, is what we're going to talk about here. And the enzyme complex that does this is the pyruvate dehydrogenase complex. So pyruvate, which of course is a product of glycolysis, and glycolysis takes place in the cytosol of our cells. Pyruvate is then transported into the mitochondria of our cells through a transporter called pyruvate translocase. This pyruvate will then enter into the citric acid cycle, which occurs in the mitochondria. But as we mentioned, it must first get converted from pyruvate into a molecule called acetyl-CoA by a large enzyme complex called the pyruvate dehydrogenase complex. And so this is what we're going to focus our attention on in this video lecture. So let's talk a little bit about the mitochondrial structure real quick, since this is where it takes place. The mitochondria has two membranes. It has an outer membrane, which is fairly permeable, and then an inner mitochondrial membrane, which is much less permeable. So that's where our pyruvate translocase is located at in order to get our pyruvate into the matrix of the mitochondria, and the matrix of the mitochondria is the innermost portion. So our citric acid cycle is going to occur in the matrix of the mitochondria, and that's also where our pyruvate dehydrogenase complex is going to be. So let's talk a little bit about this complex. We say it's a complex because it's made up of actually three different enzymes. The pyruvate dehydrogenase component is one of those parts of the enzyme, which we often abbreviate as E1. And it has a prosthetic group. A prosthetic group, again, is some non-amino acid organic molecule or organic group which helps the enzyme carry out its function. So the prosthetic group that we're going to look at that's bound to the E1 portion is called TPP. There's also, our pyruvate dehydrogenase complex also has an enzyme in it called dihydrolipoyl transacetylase, which we'll call the E2 component. And it has a prosthetic group called lipoamide, which helps it carry out its catalytic function. And finally, there's an E3 portion of our complex, which is the dihydrolipoyl dehydrogenase, and it has a prosthetic group called FAD, which we'll talk about a little bit later. So we have three enzymes that make up our complex, and each enzyme has its own unique cofactor to help it carry out a specific part of this reaction, this multi-step reaction. So this is what we're going to focus on and look at this in a bit of detail. Here's a little cartoon of our pyruvate dehydrogenase complex, just showing how large it is. If I flip back here, one thing I skipped over here is that this E1 portion is not a monomer, it's not a dimer, it's actually made up of 24 different polypeptide chains. So it's a huge enzyme, as is the E2 portion, and the E3 portion is made up of 12 different polypeptide chains. So this is a massive complex. So you can see here just each one of these circles or spheres represents one of our polypeptide chains, making up either the E1, the E2, or the E3 portion. So let's start off our reaction. Remember, pyruvate comes into our mitochondria from the cytosol into the matrix of the mitochondria. And pyruvate, if we look, of course, it has 1, 2, 3 carbons. It's a 3-carbon molecule. And when it enters into the matrix, it enters into the E1 portion, the active site of the E1 portion of pyruvate dehydrogenase complex first. In the active site of the E1 portion, it undergoes first a decarboxylation. In other words, we're removing a carbon in the form of carbon dioxide here. So that converts this pyruvate into a 2-carbon molecule, which then will undergo another oxidation step, again in the E1 active site. And then it will get transferred over to the E2 portion, where it will bind with a larger molecule called coenzyme A to form acetyl-CoA. Now again, that's just a real brief overview to show that it occurs in multiple steps, but we'll get into each of these in a bit more detail here in just a minute. So let's look a little closer at the reaction that occurs in the E1 portion of our pyruvate dehydrogenase complex. So again, the E1 portion is also called pyruvate dehydrogenase, and it catalyzes the first step. And we said it uses a coenzyme or prosthetic group, same thing, called TPP. TPP is short for thiamine pyrophosphate, meaning it has 2 phosphates on it. And we don't need to memorize the structure of this, but it does have a reactive portion right here that we'll see will help catalyze the decarboxylation of our pyruvate. Okay, so here's thiamine pyrophosphate, and here's that first step that we were looking at. So pyruvate comes into the active site of the E1 portion of the pyruvate dehydrogenase complex. Here you can see that reactive portion of TPP. So this is my TPP, but we're just showing you the reactive portion. We get a nucleophilic attack here, and what we end up with is we get an addition reaction. So our pyruvate actually covalently bonds to the TPP. We don't need to know the mechanism here, but we do need to appreciate the steps that are going on. So we get this addition reaction, and then in the process we get this CO2 breaking off and being released. So the first step, pyruvate enters into the E1 portion of our pyruvate dehydrogenase. It reacts and binds to the TPP and undergoes a decarboxylation. So we lose one of our carbons. What that forms is here, this is what we're concerned with, is what's called a hydroxyethyl TPP intermediate. Now that seems like a big name there, but of course hydroxy means we have a hydroxyl group. Ethyl, methyl, ethyl, so we have 2 carbons, so that's hydroxyethyl. And then it's bound to TPP, so we have a hydroxyethyl TPP intermediate after our decarboxylation is complete. So again, this is occurring in the active site of the E1 portion of my pyruvate dehydrogenase complex. It reacts with the TPP. We get an addition reaction and then a decarboxylation. We end up with this hydroxyethyl TPP intermediate, which is covalently bonded to our enzyme. So that's step 1. Now, the next step is going to be catalyzed by our next coenzyme, or our next prosthetic group, which we said was lipoamide. Lipoamide is covalently bonded to the E2 portion of our enzyme, but if you notice, it has a long chain here. So the lipoamide actually bonds to our enzyme. So this would be the polypeptide chain of our E2 portion, and here is a lysine residue. You can see the lysine residue here, and our lipoamide is covalently bonded to lysine. So it basically creates this long arm where we have here the reactive portion, this disulfide bond between 2 sulfurs. This is the reactive portion of lipoamide. And this long arm is important because this long arm will allow it to actually swing from the E2 portion over into the E1 active site. So we'll see that here in just a second. Okay. So here we have we're looking at the active site of the E1 portion where we have the hydroxyethyl TPP intermediate, and here comes that lipoamide. It's going to swing, that long arm is going to swing from the E2 portion into the active site of the E1 portion of our complex. What's going to happen is this hydroxyethyl group is going to get transferred over to the lipoamide. You can see that here. So we get this TPP, and then we have what's now an acetyl group because we undergo an oxidation step here. So we had a hydroxyl, now we have an oxygen double bond here. So we underwent an oxidation. So this is the lipoamide arm that has now bonded to that acetyl group in the E1 portion, in the E1 active site. So for step 3 in our reaction, we're going to transfer our acetyl group, which we have here, to a molecule called coenzyme A. So what happens is this acetyl-lipoamide intermediate swings from the E1 portion active site, and it swings into the E2 portion, its active site. In this E2 portion's active site, we have coenzyme A is loosely bound there. It's not covalently bonded. And through a reaction, we have our acetyl group being transferred to coenzyme A. So we form acetyl-CoA, acetyl coenzyme A, and then this molecule, which is the important one, that's what we wanted to form in order to go to the citric acid cycle, this molecule is able to diffuse out of the E2 active site and enter into the citric acid cycle. So essentially we're done, but there's one very important thing. These are enzymes, and enzymes have to be regenerated. So in a minute, we'll look at the last step and see how this reduced form of dihydrolipoamide is going to get oxidized back to lipoamide. Okay, but first, a little bit more about coenzyme A. Okay. Coenzyme A, it's a fairly large molecule, and we don't need to appreciate the whole structure of it, but you can kind of see that here. And it has this reactive sulfur group, and that's where our acetyl group is going to get transferred. Now, acetyl-CoA is actually a high-energy compound. We talked about high-energy molecules like ATP and others in glycolysis. But what makes acetyl-CoA a high-energy compound? Well, here is acyl-CoA, and here's acetyl-CoA. You can see acetyl-CoA, we just have a methyl group here. What makes this such a high-energy compound is that we have what's called a thioester. Okay, a thioester is basically what you can see here, where we've got a carbon double bonded to an oxygen and then a sulfur. Whereas a regular ester, if we had an oxygen here, that would be an ester. So a thioester, thio meaning sulfur, is just we've replaced that oxygen with a sulfur. So what makes thioesters such high-energy compounds compared to maybe regular esters? Well, if we look up here up top, here we have a regular ester. Okay, this is an ester functional group, and these are able to undergo resonance. And we know that resonance structures will stabilize a molecule because they kind of spread that electron density out over a larger area. So an ester is a fairly stable molecule, stabilized by resonance. But because we've got the sulfur here, we don't form these resonance structures. So that makes this a higher energy molecule. And as we'll see, when that high energy acetyl-CoA enters into the citric acid cycle, it's broken apart, and a lot of that energy is released in order to drive that citric acid cycle in a spontaneous direction. So again, a very important high energy compound is acetyl-CoA. Appreciate what makes it a high energy compound. So let's get back to our reaction here. We basically made acetyl-CoA, so we're almost done. We just have to regenerate that lipoamide. So here's where we left off. That lipoamide, which is the prosthetic group in our E2 portion, is reduced, meaning it's gained electrons. We need it to get oxidized, so we need it to lose electrons. So this lipoamide, again, with that long arm, it swings into the active site of the E3 portion of our complex. The E3 portion has a prosthetic group called FAD, or flavin adenine dinucleotide, which is an electron acceptor. Okay. So our FAD will accept 2 electrons and 2 hydrogens. So those will be spontaneously transferred to FAD, creating FADH2. So this would be the reduced form of dihydroxylipoamide. When it loses the electrons, it becomes oxidized back to lipoamide. This is my oxidized FAD. This is the reduced FAD because it's gained electrons. Now we also have to reduce or get those electrons out of FAD, H2, so it can regenerate to FAD. because FAD is covalently bonded to the active site. So there is a NAD plus molecule. We've talked a little bit about that in glycolysis. NAD plus, which is not covalently bonded, will diffuse into the active site of the E3 portion. Those electrons are transferred to NAD plus as a hydride and H minus, creating NADH. So therefore, we have regenerated our pyruvate dehydrogenase complex. We've regenerated the lipoamide, the FAD, and we're ready to go through another round of reaction. So just a little bit more about those important electron acceptors like FAD and NAD plus, because those are going to be important as we move along. We have FAD, you can see that down here, flavin adenine dinucleotide. So we have, here is our structure, and again, it's a fairly complex molecule. Up here, this is going to be our reactive portion up here. I don't know if you can see it in red here, but this is where the electrons and those hydrogens are going to be accepted. There's another molecule we're not really talking about here much, but another important electron acceptor that we may come across later is called flavin mononucleotide, FMN. Again, this, very similar to FAD, can accept electrons and hydrogens, and these are derived from the vitamin riboflavin. Now, here you can see our reactive portion of the FAD or FMN, because they're very similar. They can accept one electron to form what's called a semiquinone, a radical, and then they can accept a second electron and the hydrogens here and here. So basically, the important part here, we don't need to memorize those structures, but they can accept two electrons, one at a time, and also two hydrogens. And those electrons are not going to be wasted. Those electrons are going to move to NAD plus to form NADH, and ultimately those electrons are going to be transferred to our electron transport chain to help generate ATP. And here you can see NAD plus, its structure. Here we have the reactive portion up here, and it can accept a hydride ion, which is of course an H minus, a hydrogen with two electrons, in order to give us the reduced form NADH. So let me go through, there were a lot of steps here. Let me try to go through this one more time to try to sum it up, hopefully help you a little bit here. So this cartoon here, this is my pyruvate dehydrogenase complex. We have our E1 portion here in yellow. The E2 portion, we're showing two of those. And here's the E3 portion. The E1 portion has TPP as the prosthetic group. The E2 portion has the lipoamide, and you can see that with that long swinging arm here. Hopefully you can see my cursor moving around. And then the E3 portion, we have FAD, which is our electron acceptor. So let's start right here. In step one, my pyruvate comes into the E1 portion. It reacts with the TPP, and we get a decarboxylation, and we form our hydroxyethyl TPP intermediate, which you can see here. That occurs in the E1 active site. Now, the next step, the lipoamide is going to swing into the E1 active site, and my hydroxyethyl intermediate is going to bind to that lipoamide, forming an acetyl group. Okay. So now we've transferred that hydroxyethyl group to our lipoamide, and that transfers as an acetyl group, and then that lipoamide arm then swings back into the E2 active site. So you can hopefully see it swinging back down there here. And in the active site of the E2 portion is our coenzyme A. That acetyl group then transfers to that coenzyme A to form acetyl-CoA, and that goes into the citric acid cycle. Then we have to get everything oxidized back. So the reduced lipoamide arm swings into the E3 active site. The two hydrogens and those two electrons are transferred to FAD, giving us FADH2. So my lipoamide arm is oxidized back to where it needs to be. And then the two electrons and one hydrogen are transferred to a mobile NAD+, which then can leave and go into the electron transport chain, forming NADH, and we regenerate the FAD. So that's our cycle there. I want to be sure we understand the steps of this very important enzyme complex. Now, once we have our acetyl-CoA, this is in our matrix of our mitochondria, it can then start being reacted with a bunch of, or reacted through a bunch of enzyme catalyzed steps into the citric acid cycle. So we're going to pull off as much energy as we can from there in the form of electrons mostly, and those electrons are going to be used ultimately to help us produce ATP. So we'll get into the citric acid cycle next time.